Hello everyone, we are going to talk about a webinar Three-Dimensional Cell Culture, the missing link in drug discovery by students of Tecnológico de Monterrey. Lack of clinical efficiency and unacceptable toxicity are two of the main causes of drug failures during development. High cost, typically $1 billion in getting new drugs approved, and the fact that many oncology drugs fail during clinical testing, especially during phase 2 and 3. It is therefore necessary to improve in vitro cell-based testing methods for a more informed prediction of drug candidate efficiency and safety, and thereby save out poorly functioning components while prioritizing promising candidates. Well, it is important to know a little background of this topic. For this reason, I'm going to talk about two-dimensional cell culture. What happened with this technique? Well, it involves cells are typically grown as monolayers on a flat surface in culture flats or petri dishes with medium as a source of nutrition at body temperature 37 degrees Celsius. This technique is very used in many researchers. However, this presents many disadvantages in contrasting with the model in vivo, such as cell morphology, polarity, receptor expression, oncogene expression, and interaction with the SCM or extracellular matrix.
Extracellular matrix is a protein and carbohydrate network outside the cell. It helps with the interactions between cells, and it has different components like collagen, proteoglycans, integrins, and fibronectin.
In order to develop reliable analysis regarding drug discovery, it is necessary to create similar environments to those where the drugs will be administered in preclinical testing. 3D culture allows this since it uses functional tissues instead of single cells. For example, mammary tissues comprises epithelial cells, which present a specialized cell-cell contact interactions, a polarized morphology, and are attached to an underlying basement membrane. The preservation of these features is essential for normal function of the tissue, including proliferation, differentiation, survival, and secretion. For example, it has been demonstrated that the functional unit of breast tissue includes not only the cells, but also the extracellular matrix where they interact with each other. Therefore, the use of 3D culture allows the cells to retain certain characteristics and the possibility to incorporate elements to the extracellular matrix. The main differences between both methods are: first, in 2D culture, the cells are usually grown as monolayers, and they require to be attached to a surface in order to survive. Although cells that are cultured this way can retain some properties from the in vivo environment, most of the interactions and signaling pathways may be lost or compromised. On the other hand, 3D culture allows the cells to mimic better the in vivo tissue by growing them in three-dimensional shapes, such as spheroids. In contrast with two-dimensional culture, this one actually requires the cells to be not attached to any surface, but among them. Regarding the interactions and signaling, 3D culture actually preserves cell-to-cell contact and interactions between cells and the extracellular matrix, which is essential for biological processes such as proliferation and differentiation.
Forged floating methods. This technique works under the principle that 3D spheroids are formed by preventing their attachment to the plates. This is obtained in two ways. You can either buy pre-coated plates with a special treatment, or you can coat in your own lab the plates with agar or polyheme. In this technique, cell-cell contact is promoted as well as multicellular sphere formation. The size of the spheres can be adjusted, plus the cells can get a larger sphere. One of the main advantages of this technique is that it is inexpensive when you are using agar. You can also have an homogeneous result when researching for drug toxicity and gene expression in spheroids. However, as any other technique, this also has its own disadvantages. Agars requires an additional step for sterilization, autoclaved before coating the plates. And on the other hand, polyhema takes several hours to dissolve in 95% ethanol.
Hanging drop methods. You need to place an aliquot of 20 microliters of a single cell suspension, so this will be pipetted into wells. After the sealing, the tray is inverted and the aliquots turn into drops, and the cells accumulated at the tip turn into 3D spheroids. Now, the advantages are that it is simple to do and 100% reproducible when producing one spheroid per drop. There is low variability in sizes, and matrices and scaffolds have no effect on the 3D structure. Now, there are some disadvantages. The volume of the drop is limited, a maximum of 50 microliters, because with a larger volume the surface tension can be broken, and also the culture medium is difficult to change, meanwhile not disturbing the spheroid. In the case of the agitation-based approaches, the principle is that cell suspension is placed into a container and kept in motion to avoid cell adhesion to the walls. The spinner flask bioreactors use a container and a steering element, and in a rotational culture system, the culture container is rotated. The advantages of using a spinner flask is that there is control over culturing. The medium can be changed frequently to have long-term 3D culture, but the disadvantages are that cell physiology can be affected. Large medium requirements and different sizes in spheroids are obtained. In the case of rotational culture system, the advantages are that there is low shear force, it is a simple method, and a large-scale production of 3D spheroids can be done. But the disadvantages are that you need specialized equipment, and the number of cells produced can't be controlled. This is why manual selection is needed.
The matrix could be a nutritious gel. Enriched with proteins and growth factors, it mimics under in vivo conditions the MCM, allowing cell-to-cell interactions. The matrix provides nutrients for cell differentiation, so cell spheroids should develop and resemble to the original organ or tissue. The main disadvantage is that the MCM is a biological substance, so every batch differs from another. In addition, large-scale production might be expensive, and it presents a random size and location of the spheroid in the media.
Prefabricated scaffolds can alternatively be used to a 3D culture. Cells seated into such scaffolds can migrate between fibers of the scaffolds and attach to them. The interstitial space between fibers is filled by cell as they grow and divide, forming 3D cellular structures. Collagen, laminin, alginate, and other biodegradable materials are commonly used for this purpose. These products can be processed to form hydrogels for use as 3D cell culture scaffolds. Gels that incorporate ECM molecules can apparently communicate in vivo-like cues to cells to enable in vivo-like cell behavior. The size of pores in a hydrogel can be manipulated as necessary during production, enabling hydrogels to be engineered to suit experimental requirements. Hydrogels can also form the base for cell-compatible microcarriers that enables a large number of cells to be cultured at any one time using small volumes of medium. Microcarriers can be coated with cell adhesion promoting substrates. These microcarriers have the advantage of a hydrogel form that alginate core and have the ability to culture large cell numbers due to their large surface area. The downside to these carriers is the requirement of specialized equipment.
The microfluidic cell culture is a system that comprises a main microfluidic channel, which is an array of micropillars through which the cell suspension is passed. Also, this micropillar system immobilizes the passing cells and it supports their growth. Also, enables cell-cell interactions. The advantages of this method is that it has a potential for high throughput drug testing and it can be compatible with high content analysis. And also, quantitative and quantitative analysis are enabled. However, the disadvantage of this method is that there is no chance of doing recovery of the spheroids and their characterization. And also, some analyses like the immunoblot are difficult to do because the sample has small volumes.
Then I will explain an application applied for the company Merck. It is a pharmaceutical company important around of the world. This applies a 3D skin culture as an in vitro model of the human epidermis. Historically, animal models have been used to test the toxicity and efficiency of cosmetics and transdermal drug absorption. Due to ethical concerns, however, these animal models have gradually been replaced by cruelty-free in vitro organotropic skin models that use primary human cells and cell culture inserts to recapitulate the stratified epidermal architecture. For example, in this picture shows the procedure used for this aim. In this procedure, use different cell lines, for example, keratinocytes, more fibroblasts.
Now that the different methods have been described, it is worth to mention the importance of 3D culture for further applications. For drug discovery particularly, there is enough evidence on how the microenvironment affects the response of the cells to therapeutic drugs. Therefore, some points must be considered for future assays regarding drug testing. First, it could be convenient to run 2D and 3D culture simultaneously, in order to compare responses and different behaviors of the cells when they are exposed to different drugs. In the case of cancer, 3D cultures should be used in order to test novel drugs, because this way similar environments to in vivo tumors could be created. And the last but not the least, it is necessary to consider that 3D cultures can actually generate and yield more reliable results on the uptake and the distribution of the drugs inside the cells, which may be very useful in order to determine as early as possible if it is worth to continue or not with the development of certain drugs.
Conclusions. Nature is perfect, and cells should be developed naturally in a 3D environment. This must be the main objective to mimic during tissue culture. Second, 3D culturing affects proliferation, morphology, differentiation, and cellular functions. Continuous application of 3D culturing would make the process of drug development more cost-efficient. And although 3D culturing is very promising, it still needs optimization. New ideas and methods should always be taken in consideration in order to achieve progress.